I'm Sean Davis. It's my real pleasure to introduce our speaker this morning, Corrie Painter. She and I met about a year and a half ago in a session that was developed around the idea of genomic data sharing and how patients benefit from and don't benefit from getting access to and having control over their own data. And Corrie has been a real leader in thinking about how data and patients and research are really sort of partners in this process of trying to understand this horrific set of diseases called cancer. So without much more detail than that, Corrie is the Associate Director for the Count Me In project. She's a researcher scientist at the Broad and Harvard. She's a trained cancer researcher with a PhD in biochemistry. And she's actually she was doing her postdoc in cancer immunology, focused on melanoma. At around that time, she was diagnosed with a rare form of cancer called angiosarcoma. And she's combined her cancer advocacy, scientific background, and community building to engage with patients in order to build and carry out patient-partnered genomic studies. And she's also a very active advocate and is the co-founder of the Angiosarcoma Awareness Incorporated. So without further ado, let's turn it over to Corrie.
Thanks so much, Sean. And I'd like to thank the organizers for inviting me to come present to all of you today. And I'd like to thank each and every one of you for your attendance. And I hope you're all well and healthy and weathering these times as best as possible. So Sean gave a very generous introduction, and I'm going to expand on some of the things that he talked about in terms of setting the stage for why I'm so interested in this notion of data sharing and partnering with patients. I kind of feel like I have to provide some context with my personal story around all of this. And so it's going to be pretty personal, and it's going to, weave in and out of science plus my own cancer journey throughout this talk. So as you think of questions, please know that I'm a very open person and happy to answer anything, whether it's about my own personal experiences, advocacy, the work we're doing at the Broad Institute, or beyond. So, I was diagnosed just over 10 years ago with this cancer that Sean had mentioned called angiosarcoma. I was a grad student at UMass Medical School getting a PhD in biochemistry and had a lump in my breast. And it was something that the last thing I would have thought was that this was going to be an exceedingly rare and aggressive cancer that had a very poor prognosis. But after a challenging 4 months that consisted of several biopsies and surgeries and diagnoses and misdiagnoses, I was told I have angiosarcoma, which is a cancer that starts in the inner lining of your blood vessel, the endothelial cells. You can think of your blood vessels as a hose, and the very inner lining of that hose are endothelial cells. So you can imagine that your blood supply can feed the cancer very readily when it is the actual lining of those blood vessels that is the malignant component here. So this is a picture of my husband and I. He, in solidarity, had shaved his head. He looks like the cancer patient, and I'm waiting for a scan. But this picture really represents what the first several years of this was like. And my kids were 2 and 4 at the time. I'm actually looking over the top of my computer right now at my now 15-year-old in amazement that we've come this far. When I was diagnosed, I was already a scientist, and so I understood how to look through the literature. First thing that I did was go to PubMed, and what I found were these just retrospective studies that had very low numbers. They were trying to draw statistics from 14 people that may have been treated at a single institution over the course of 60 years. There were single case studies talking about one patient's clinical experience with the disease, and one of the papers was actually entitled, evidence of long-term survivorship in an angiosarcoma patient that lasted an entire year. And, this is very disheartening when you're the scientist reading this. This is not what you want to see at all. And so I decided, I was not going to be able to learn anything scientifically about this, but that maybe I could find some practical experiences that would help me. Maybe there were other people who had gone through this who were also looking to connect, and we could form some type of nucleus of understanding and share experiences and learn from each other. So I went to Google and I would type in angiosarcoma cancer, and sure enough, there were people, they were also looking, and I'd find them and I'd reach out to them, but not one person responded. And after, some time, it occurred to me that maybe they're not alive anymore. And so I'd take their name and I'd Google search it, and lo and behold, their obituaries would pop up every single time. And again, very just, it was just a time of despair, as you can imagine. So I went to Facebook, and this was 10 years ago, so it wasn't quite the same social media platform that it is today, but it was still very robust, and it was a place where people were connecting with each other. And in a last-ditch effort to find anybody, I typed in angiosarcoma cancer there, and lo and behold, there was a group started by this woman, Lauren Ryan, with about 10 people in it. And I instantly connected with these people on a level like a friendship and a kinship that I had never experienced before. These people understood every single thing I was facing. And they were stay-at-home mothers, they were artists, one was a cop, they were just a spectrum of people living their lives who got slammed by this diagnosis just like I did. And they were just trying to connect with each other to share things that they thought might be useful. And they were so useful. And though I knew it wouldn't lead to scientific insight, it led to kind of a life raft in the middle of a hurricane. So Lauren, who founded that group, and Lauren was a stay-at-home mom, decided that we were going to launch our own nonprofit together. So we co-founded Angiosarcoma Awareness. And the purpose of this was to fund research labs that were engaged in vascular biology, if not angiosarcoma directly, though because it's so rare, almost nobody was studying it. We came up with our own mechanism. Because I'm a scientist, I could understand what each of the labs could offer, and then maybe kind of tether different labs that would never otherwise find each other through a funding mechanism. So instead of trying to go through a normal RFA, somebody writes a hypothesis-driven set of experiments that they want to do to me, and then we give funding. It was almost turned on its head, where we would look for different labs that had the capability of doing specific types of research, and then other people that may have resources not available to the other lab, and talk to both and have them co-design a grant together and then fund both. And we tried to do this in a way that would maximize the ability for data sharing, integration, and collaboration. And at the same time, we were also engaged in peer-to-peer advocacy and trying to make savvy patients in the space. These efforts led to over $750,000 within 4 years, funding a bunch of different labs with that notion of really trying to open source the information that they generated. So if they were successful in generating a xenograft mouse model, make it available to people. If they generated a data set, open source it so other people could use it and leverage it for their own knowledge base. And that was the hope when I was going through this. So in order to push that advocacy forward, I hadn't graduated yet. This all went down 2 weeks before I was supposed to defend my thesis. In fact, there was a chair with my name on it at commencement. I was not in it. So I wasn't sure at that time if I was going to live, and if I was going to live, for how long. And I just, my PI at the time, my graduate student PI, said to me that he would be happy to keep me on forever as a graduate student, knowing that forever was probably 6 months until I was no longer going to be alive. So he was very generous with keeping me on throughout the duration of this disease. And I felt no pressure to actually defend that thesis until I was not getting responses from scientists when I would reach out to them as Corrie, the cancer patient. So it was a very risky move, but I decided to defend my thesis. And it was risky because I had to go off of my student Blue Cross Blue Shield insurance, which was top of the line, and go on to postdoc insurance, which was bottom of the barrel. But it was worth the risk to try and push the advocacy forward. And so I defended that thesis in June of 2011. Lo and behold, a year later, I was still alive, and it was remarkable to still be alive. And I thought to myself, well, maybe I do have some time here on this earth, and maybe I can do more with whatever time I have left. So I went ahead and I wrote for a grant and received funding to do a fellowship in cancer immunology studying melanoma. And those studies went really well. And I had the opportunity in front of me to be my own, PI, to have my own lab, or to push the forefronts of advocacy forward. There was a faculty position offered to me for tenure track, but at the same time, if I did that, I would have had to step down from my role as an advocate. And I thought to myself, what I could do with my own lab pales in comparison to what I might be able to do by inspiring many, many labs to work together at once. So I decided not to take any faculty position, and instead looked for just kind of a normal, quote unquote, normal job that would allow me the time where I could do both, where I could be a scientist during the day and do all of the advocacy at night and on the weekends. At the same time, the Broad Institute had posted a job position for the Associate Director of Operations and Scientific Outreach in their cancer program. And when I read that description, it was as though somebody looked into my life and made a job based on my crazy experiences. They were looking for somebody who understood what it was like to be a cancer patient, who understood the world of advocacy, and who was a PhD level cancer research scientist. And they wanted this person to conceive of a new infrastructure that would engage with cancer patients in order to generate massive scale genomic data and other types of data in order to open source it. And I thought, my goodness, this is nothing has ever been this aligned in my life. And so I jumped at the opportunity. And what it looked like from the perspective of the folks at the Broad who were leading these efforts. So this was kicked off really from the Broad leadership and led by Nikhil Wagle, who's now the director of Count Me In, which is the suite of projects I'm about to talk about. And the notion was this, that there are so many challenges with studying patient tumor samples that exist right now in our country and in the world. And prevalent among those is the fact that 85% of cancer patients are treated in community settings. I'm one of them, right? My tumor was biopsied at my local medical school, which actually has a cancer program, but they don't study angiosarcoma or sarcomas at all. And so my biopsy went into a ball of wax. It was used to make my very first misdiagnosis, and then it sat on a shelf, and it was not leveraged to understand this disease at all. And that's the same kind of course that most of our tumor samples throughout the country take. They're used for the diagnosis, and that's the end of the line for them. As well, in addition, our experiences, what I can talk about from the vantage point of being a patient is not incorporated into the fold of how we conduct research at all. So most patients' tumor samples and their data just haven't been readily available for studying. But we live in an era now where we have technology and social media, and there have been cultural changes that enable us to really think about partnering with patients in a way that was never possible before. Now, I'm going to date myself with how old I am. I'm not 20 like I look. I'm in my mid-40s, and when I was growing up, people didn't talk about cancer, and when they did, they talked about it in hushed tones. But now it's kind of become part of the national dialogue, and it's not as stigmatized in certain populations as it used to be, and that's a very thick underscore in certain populations, because there are still so many issues that we from the research side have to confront if we're going to destigmatize this across all patient populations. But suffice it to say, we are in a place now where we can at least begin this process of talking and working directly with cancer patients. The hope is that we can generate a massive publicly available database of clinical, genomic, molecular, and patient-reported data in cancer to enable researchers to find patterns in the data and to accelerate discoveries in the development of new treatment strategies. That's the hope. How do you do that? Well, step one is talk to lots and lots of patients directly impacted by the disease you're trying to study if you want to partner with them. It's not necessarily the first go-to that research scientists have, like talk to the end user of whatever it is that you're trying to do. But when you're trying to partner with patients, it's really important to talk with them in the lead up and build of the study that you want to engage with them in. And in this case, we were going to launch our project in metastatic breast cancer. So Dr. Wagle, working at the Dana-Farber Cancer Institute as a medical oncologist, knew several patient advocates, and we started our conversations there, where we asked them, what should we name the project? What should it look like? Should there be a picture of somebody with the disease? What kind of language should we use? So we were asking the most basic questions of these advocates to get a sense for where do we even start. And we took the information that they gave us and we then brought it into Twitter, where we asked more metastatic breast cancer patients to help us build this project. And they did. You know, we had dozens of patients that worked on every single part of this project. They helped us realize it, you know, first of all, we should not try to build a community, that there are already communities that exist, and rather we should be creating something like a website or an app that can be brought into the communities that already kind of cropped up organically. And that we should use the word metastatic breast cancer in the title of the project, that it would be a learning experience just by virtue of the name itself. They asked us to make the burden of participation as low as possible so that they were not, you know, put under undue stress trying to help. And so they helped us both message this, they helped the imagery that we used, and then they helped us develop and design the operations on the back end for like, what does the patient do if they want to join, and what does our study staff do after a patient has joined? And with their help, we launched this in October of 2015. And to date, over 5,700 women and men from across the country and Canada have said, count me in. I want to be part of this. And I want to leave my legacy in order to make sure that other people don't have to go through what I'm going through right now. I'm going to talk a little bit about what enrollment has looked like over time. And it's been really phenomenal. We launched in 2015, and when we launched, we engaged with about 6 different grassroots organizations that all had patient reach into the metastatic breast cancer world. We specifically wanted this narrow focus of people who had like patient touch in order to help us refine the message and connect deeply with the community. So we launched, and the launch is basically like, now the website is available, but we launched with some messaging that was also promoted by our advocacy partners. And lo and behold, you know, having no idea what this would look like, we had almost 800 people join in that first month. And it was remarkable. We were, you know, we honestly had no idea what to expect. Would 100 people join? Would 1,000 people join? So having 800 people within 1 month join any study it's pretty remarkable in terms of the amount of learning that you can do. So when it started to tailor off, you know, toward the holidays, we were, you know, our opinion of it was that this is really good. And maybe we've captured everybody who's going to be involved with this. And if that's the case, we're still going to learn so much. And so we were super grateful for the people that had joined, and we were ready to you know, start generating that data. But then something remarkable happened. We presented at a conference called the San Antonio Breast Cancer Conference in 2015, and we got a bump in enrollment. And then about a month later, we saw this really large spike where a couple hundred people joined within 24 hours. And we were able to find out that this was entirely due to one patient who learned about the project. And she was a trusted kind of cornerstone figure within the patient community. And she posted about this project. And many of her followers, hundreds of her followers, decided to join. And then they started to share about it. And so you see this steady state enrollment over time with these stochastic jumps that are really brought about by either press or a prominent patient that's you know, talking about this in social media. But most of this is word of mouth from one patient to another in their Facebook posts or in their Twitter feeds, and really kind of driving our ability to reach patients forward. To date, we have over 2,000 medical records that we've received from patients. We have 586 tumors from 425 patients. We've completed whole exome sequencing on over 100 of the tumor saliva matched pairs, and we have almost 1,000 blood biopsies in-house. So when a patient joins, and if they provide consent, that enables us to get a portion of their archival tissue, that same tissue that I had taken out of my tumor and that just sat on a shelf. We can get that now from patients who give us their consent. And we need to look at the DNA mutations in that relative to that patient's normal DNA. So we ask for saliva. And in some cases, we also can get blood samples, which both can provide that normal DNA from the white blood cells, but also may contain bits and pieces of shredded DNA from inside the tumor that we can actually sequence through whole exome sequencing the same way we could from that person's actual tumor. And so it could give us a glimpse of the mutations that are arising in tumors throughout a metastatic patient, just by virtue of taking a blood draw, which is really remarkable. So this is a glimpse of what this looks like in social media. Everybody in these posts, except for this woman here who is a phlebotomist and this woman here who's a nurse, is a metastatic breast cancer patient. And again, men get this disease too. So you can see our friend Bob over here in the top left going through treatment and sharing a picture of his kit. These people are sharing their saliva or blood kits, and they're smiling. These are terminally ill patients, 3 of whom have already passed away, that were smiling at the point of, being able to participate in this research project. And they were smiling for their own reasons, and they were sharing those reasons in social media, encouraging other people to join them, and they use their own experiences and they use their own language. And I think it makes it just so much more authentic to have it an organic movement being led by patients rather than research scientists going and trying to accrue through the normal traditional mechanisms. And it's what's led to that steady state enrollment over time. One of the other benefits of doing a decentralized study where anybody can sign up just on their cell phones or their tablets or their computers is we can engage with patients who are scattered all over the place, who may not live anywhere near a major academic institution that is studying their disease. And you can see here in this graph, we have patients that are being treated at over 1,700 institutions. We see some of the very large institutions that have dozens of patients, but there are over 1,200 institutions across the country that have one patient that's participating. I can't imagine a mechanism where you'd be able to build a consortium from a top-down project. This type of response can only be initiated if patients are directly involved and using their consent and their will to drive this research forward. So we want to take all of the data that we're gathering and generating and stitch it all together so it's linked in order to generate that open clinico-genomic database that I had talked about in the beginning. And so far, we've been able to generate data and release it into cBioportal and the Genomic Data Commons. We also want to release it into other research portals. This is right now actually Terra, not Firecloud. That's an old slide I need to update, at the Broad Institute. And we want to generate patient and researcher specific portals. And I think I really want the folks in this particular conference to really think about what might the data look like for computational biologists. What could we do? What could we develop that would maximally leverage this data in order to share it as broadly as possible? And if you have ideas, I would love to hear from you. So find me at painter@broadinstitute.org and share them with me. I would love to hear your thoughts. So right now, if you go to cBioportal and you look for the Metastatic Breast Cancer Project, or if you just go to our project website, which is mbcproject.org, and you click on data, you can access this on cBioportal. This has all been de-identified, and it's only the somatic whole exome sequencing that we're releasing here. So we can do this without password protection. Anybody can download it. It's been stripped of all readily identifiable information and shared in aggregate. So you can, take a look at the data, and it's got very robust clinical fields. If you do go to cBioportal and take a look at this, make sure you go to Add Chart and click on that downward icon, and you'll see about 80 different clinical charts that pop up. And what that enables you to do is it enables you to filter on the data to build whatever cohorts you want in order to understand the associated genomics that go along with it, and or in the inverse, find the genes of interest and see, does it associate with young people or HER2 positive cancer or sites of metastasis to the bone? And in these ways, you can generate hypotheses and or augment your own understanding from the research that you're doing in this disease. So we weren't sure, again, when we released the data, would it be used? And sure enough, within the first 6 months, over 5,500 downloads from cBioportal took place. And to date now, there's over 20 publications that are citing this data. And to my point, originally about, should I be my own PI, or should I try to inspire other people to do research? Which one will have more impact? I can say emphatically now, the latter. There's no way one lab within a couple of years of sharing data would have 20 publications and more coming every day. And so I feel like, for those metrics, for the amount of interest in the patient population, the fact that folks are generating publications that are in peer review and making discoveries and sharing those more broadly, that these are metrics of success for this whole way of conducting research. I'd like to share a couple very short videos so you can hear directly from patients what they think of the project and what they think of the data. The first person I'm going to introduce you to is Beth Caldwell. Beth, she was there from the beginning, helped us build the study. Though she looks great in this video, we knew she was very sick, and she died a couple of weeks after making this video. There was a tremendous sense of urgency to get her feedback on the data before we released it. So we reached out to her first and then engaged with 40 different participants within the project to go through what the data looked like inside CBioPortal to make it as patient-friendly as possible before we released it. And I have to say, no matter how long I've been engaged in this research, and though I wear the patient hat, I'm always wrong, with my assumptions of how people are going to react to different elements of the project. And it's really held my feet to the fire with respect to making sure we always do due diligence by asking people their opinions and not hoisting our own beliefs and our own assumptions onto them. When we made this data, I had my own particular set of thoughts with respect to how patients would interact with it. This was never on my radar, but Beth is going to tell you something that we heard time and time again during those 40 patient walkthroughs, and I'll let her speak for herself.
Wow. Okay, that's really cool. This is like so much information, and I feel like patients, part of being a patient is you're so alone in this, right? Like that's just sort of the isolation that comes with having metastatic breast cancer is very real. And this makes you realize that you're part of some group that is, we're all experiencing things together.
So I never thought somebody would look at genomic and clinical data and see themselves and see the humanity behind it. But this is something that so many patients articulated, that when they saw it and they realized that they were contributing to this, that they could see their living legacy. I'm going to introduce another patient who is still a staunch advocate for the project. Her name is Kelly Shanahan. And I want to also just talk about this notion of engaging patients at every possible place. When Nick and I and the rest of the team, which has grown substantially since we launched, present at national conferences or even small conferences, we always put a call out on social media, or sometimes through an email to the entire cohort, letting people know where we're going to be, what we're going to be talking about, and to encourage folks to come join us in the presentation. And so this was, I believe, at ASCO a couple of years ago. And Kelly, who is a doctor, she's an MD who had to step away from her practice as an OBGYN after her diagnosis with metastatic breast cancer, joined us. And so Nick and I were presenting, and Kelly was right there with us, shoulder to shoulder, presenting this to the rest of the clinical and scientific community at ASCO. I want you to take note in the difference. If you've ever been through a poster presentation or poster hall, I want to ask if you've ever heard somebody talk so passionately about a research project as Kelly does.
So this is the Metastatic Breast Cancer Project, and this is one of the things that I am most excited about in my life. The Metastatic Breast Cancer Project was one of the first things that talked to us, the patient, and asked us what we wanted and how we could help. And you know what? We signed up in droves. I am this little dot here on the California-Nevada border.
So just like I was saying before, Kelly is treated in a community setting, but she wants to be able to drive research forward. I mean, if you're going to have an awful disease that's going to take your life away, many people want to just like throw a dagger at it if they can and while they still can. And it's exceedingly frustrating to feel like you are powerless, not only over the course of the disease, but in your ability to help other people. And so being able to participate in a project like this has really empowered a lot of people to have ownership over the future of this disease, if not the clinical course it's going to take for them personally. So then, you know, we felt really good about the direction of the Metastatic Breast Cancer Project. And the question became, can we do this in other cancers, or is it something unique to the metastatic breast cancer world? It's a relatively, you know, common cancer compared to many, and although it's much more rare in the metastatic setting. It's got a lot of advocacy support within the community and many vocal patient amplifiers. So, but if this kind of platform could be leveraged for something that has none of those things and still work, then maybe this could be widely applicable to all cancers. So we turned our attention to angiosarcoma, which is my cancer, which has the poorest prognosis of soft tissue sarcomas. It only has about 300 people a year diagnosed. And as you can see, this is a Kaplan-Meier curve where you have survival on the y-axis. So you can see all of the people are alive at the beginning of this, but over time, and on the x-axis we have months, you can see each one of these notches is a person dying from this disease. And it's got a very steep drop off over the first 2 years. As a scientist, this is actually the very first graph that I saw, where it was one major academic institution's 40-year retrospective analysis. And it was, I actually got vertigo looking down that steep curve, because you don't, you know, the odds are not forever in your favor when you're looking at something that over the long run has less than a 20% survivability rate. So we now have a cancer. It's exceedingly rare. Not a lot of people, you know, so not a lot of people get it. You have a better chance of getting hit by lightning. So because there's so few of us, we're not treated at the same institution. We're scattered all over the place. So even if somebody wanted to study us, we'd have to all go to the same place, which is, you know, highly unlikely. Though we have actually generated something like that, which I can talk about later, for one doctor as a result of our advocacy. But suffice it to say, in most exceedingly rare cancers, we are scattered. And because it has such a high mortality rate, there's not a lot of long-term survivors. So we wanted to point the study toward this cancer to see if this would work. Luckily for us, we had the Metastatic Breast Cancer Project in order to say, this is what it looks like. Would you, in this community, which consisted of that one Facebook group that Lauren started many, many years ago, would you help us build one of these for our cancer? And so the patients within our own disease, my own disease community, reshaped the imagery, the words to speak to our patient community. And we launched this in 2017. And I have to say, there are two things that really took me by surprise. One was the reception by the sarcoma physician community, which they've just wrapped their arms around this. The second we launched, you know, there are people I know and people I don't know that took to Twitter to let everybody else know that this was happening. One of them is Dr. George Dimitri, who is the head of sarcoma at Harvard, who happens to be my personal doctor and now scientific collaborator and co-author. But at the time was just my doctor, and he took to Twitter and said, my favorite quote of all time: tell your friends, 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 and friends of friends, friends don't let friends' angiosarcomas go unstudied for progress. And this is just a sense for how much this community really embraced the study and helped to tell their patients about it, and has led to what is now another very successful instance of patient engagement in genomics research. The last thing I kind of was expecting and just was so moved by was the response from the metastatic breast cancer community. Here's Kelly and Beth again in Twitter, and another patient who is not necessarily related to the project as deeply as Kelly and Beth, but still a staunch advocate for it, asking their people in their network to become aware of this project. And rather than seeing it as kind of a competing project or taking the light off of the Metastatic Breast Cancer Project, which was still to this day moving full steam ahead, they saw it as they rightfully should. They saw themselves as kind of the very, the people that were going to open the door to this whole new world of research. And as a result, they're going to be able to help their own community, but also they're going to be able to help this other community that would otherwise have no resources available to themselves. And they took pride in that, as they should. And that just led to yet another line of interconnectedness between, our projects and the patients that were helping to drive this. So to date, it's about 2.5 years since our launch. And for this cancer that only gets 300 people a year, we've had over 500 women and men with angiosarcoma join and say, count me in, I want to be part of this. And it's not just people with the disease. We also have a separate portal with over 150 people who have filled out surveys on behalf of somebody that they knew who passed away from the disease. So this is a representation of where folks live. In orange are patients, and in blue open circles are loved ones that provided information that's been key to our understanding of this disease. It may look like there's clustering, but this is actually a perfect representation of the population of the United States. And it's more representative of the fact that wherever there are people, you're going to get some proportion of them that get each of these different cancers. So much like the Metastatic Breast Cancer Project, we were able to release data from this project. And this is more updated, but our first data release only had 19 patients. And we saw this one graph down here that you don't need to be a scientist in order to understand something is not right there. In fact, I asked my children to take a look and say, what's different here? And both of them pointed out the fact that you have a group of people over here, but then you have a whole other group of people over here, and they're different. Something's different about them. What we're looking at right here is called tumor mutational burden. And what it is it counts the individual mutations in the tumor DNA relative to what your normal DNA has. And why this is so important is that within the last few years, there's evidence to suggest that people with cancers that have high tumor mutational burdens may actually have good responses to a type of therapy called checkpoint inhibitors. And some of these have curative capabilities. So when we saw this, we were absolutely thrilled. And we wanted to learn more about that cohort of patients. You can just go into CBio portal and you can put a box around who these people are, and it returns their categories, their clinical categories. So all but one of these patients had head, neck, face, and scalp cutaneous angiosarcoma. And so it's basically the skin of sun-exposed areas. And because of this being the case, that it was sun exposed, it led us to develop the hypothesis that maybe what was going on here was that there was sun damage similar to what you see in melanoma, but instead of happening in a melanocyte, it happened in an endothelial cell. But the cause of the cancer is basically the same as melanoma. So Isha Jain, the computational biologist who's part of our team and part of Nick Wogley's lab at the Dana-Farber, ran a signature analysis of all of the samples in the cohort. What you see in yellow, which also on the y-axis here, as you increase its higher tumor mutational burden, and the colors are coded for different signatures that you can actually see and read out of the DNA. All of these people that had high tumor mutational burden also showed a UV light signature within their tumor DNA. And everybody with an asterisk here had head, neck, face, scalp angiosarcoma. So this data suggests that angiosarcoma, cutaneous angiosarcoma, is caused by the sun and may have a response to immunotherapy if it's acting similar to melanoma. So what we thought next was, if that's the case, and because this is a decentralized study with people treated in the community, what if in medical records of some of these patients, we could find people whose doctors used a compassionate care approach to giving them a checkpoint inhibitor, because it's widely prescribed now, because in a lot of different cancers and in a community setting where, doctors are so in need of kind of the next thing they're going to do to try to provide a better clinical course for their patients, that they're really trying different things. So we looked at the entire cohort of patients to see, has anybody been treated with checkpoint inhibitors? And if so, what kind of response did they have? Lo and behold, we found 6 people. 3 of them were over here. They were within categories that were where their angiosarcomas started internally. Again, it's wherever you have blood vessels is where this tumor can start. And so we found people that had it start, in an internal organ or somewhere that's not sun exposed and that did not have a UV signature. And those 3 people went on protocol, and none of them had a response to immunotherapy. I want to caveat this with the fact that I do personally know an anecdotal, one story where there was a person with an internally arising angiosarcoma that had a different marker called PD-L1, and they had a very robust signal of that, and they had an extraordinary response to immunotherapy too. So just because they don't have a high UV signature does not necessarily mean they're not going to respond. But in this situation, the 3 people that we had in our cohort didn't respond to, with their internally arising angiosarcomas. There were 3 people that had head, neck, and face angiosarcomas. One of them, with their very first dose of checkpoint inhibitor, had an extreme reaction and had to be taken off. And so we were not able to see what happened to them as a result of taking it. But then there were 2 people that had metastatic angiosarcoma that arose from this cutaneous tumor. And I'm going to show you the clinical course of just one of them, but both had a very similar story. This one patient was on a clinical trial and it didn't work. They had progression. They went on a different clinical trial and it didn't work. They had progression. They went on a tyrosine kinase inhibitor called pazopanib and it didn't work and they had progression. And then their doctor used pembrolizumab, which is a checkpoint inhibitor, and they had an amazing and complete response, meaning their tumors all went away. And then after a long time on this drug, which the drug itself kind of takes the brakes off of your immune system so that your own immune system can react and kill the tumors. But, it can also lead to some autoimmune disorders as well. And that happened with this patient. So they had to be taken off. But despite the fact that they were metastatic, even after they were taken off of this treatment, they remained without evidence of disease. And this has been updated to 2020. So it's been several years and they've had no recurrence of this cancer. In their medical record, their healthcare provider wrote this: after the first cycle of pembrolizumab, this person had a dramatic reduction in the size and mass in the neck and retrooral area. This is just amazing. And when you read this from, the perspective of our science, where we're trying to aggregate these stories and really understand what's going on, this is exactly what we're looking for. These individual needles in the haystack of responses that are happening all over the place that would just be sealed away in a tumor sample on a shelf or sealed away in somebody's medical record, regardless of how incredible that response was, can come to life now and can inform the entire community because we can now share this freely. The second we saw this data, we were giving talks within the sarcoma community. And so in advance of even publishing this, just making the data available, investigators from across the country were able to, look at the data for themselves and use it to try and develop clinical trials. And to date, there are at least 3 clinical trials that are enrolling angiosarcoma patients to checkpoint inhibitor studies to test this clinically. I want to go ahead and just highlight one quick piece of discovery that we also made, which is in a particular protein called PIK3CA. This protein also has a tremendous amount of mutations in breast cancers. And in our cohort, we found that there were mutations only in breast angiosarcomas like mine. And so we have a situation here where we made discoveries in the cutaneous cohort, and we're also making discoveries in the breast cohort that are unique to patient involvement in the build of this study. These series of mutations aggregate around where we know the breast cancer mutations, the canonical mutations are. And if you look at the way that these mutations kind of arise, you can see that there is similarity in how they may function relative to the mutations found in breast cancer. And so this is another point where maybe there could be clinical implications for building clinical trials around breast angiosarcomas that use PI3 kinase inhibitors. We were able to aggregate all of this data and publish it in Nature Medicine this year. And the response from the community was amazing. And I don't have time to read all of the responses, but I'll just read this one simple one. Miracles do happen. As a result of the success of these projects, we were able to launch several additional projects. And this map represents patients that either have, the angiosarcoma or metastatic breast cancer, but also metastatic prostate cancer, stomach and esophageal cancer, brain cancer, all comers brain cancer, and more recently, osteosarcoma. I want to highlight the osteosarcoma, which is our most recently launched project. and mostly to show you how different it looks. You know, we build these projects with the patient communities, and in this case, we built it with children. And the children wanted this, they didn't like the silhouettes, they didn't like how wordy it was, they didn't like the fact that there were no videos to explain what was going on. So we radically changed everything and pointed it toward their population and what they thought would resonate. And you can see Dylan here smiling on our homepage with a video describing what's going on, with a learn more button for those who really want to dig in more to the details of the project. We always encourage people to come visit us and to engage at every opportunity. So here are people from within the osteosarcoma community. There are parents, there are children. Our two key investigators who are leading this, Katie Janeway and Brian Crompton, were there. And we just all interacted with each other in a way that helped us each understand each other's perspectives better. This led to a revamp also in how we show what participation looks like. And the project so far has been successful with over 100 people joining within the first couple of weeks of launching. This is one of my last slides. I just want to show what it looks like to have people from different projects sharing their selfies in social media, encouraging other people to join with these testimonials. And I'll just read this one. It's so impactful to me personally. It has never lost an ounce of meaning over the years that I've been doing this. I want to live and watch my children grow up, but if I can't, I want to leave a legacy and a cure. It always chokes me up. And, you know, just for a point of levity, I just also want to point out that, you know, sociologists can also study this. You know, they can also take part in this. And I'd love their take on why it is that all the women have taken selfies, but all of the men have had pictures taken of them with their boxes. I'm not going to draw any conclusions, but I will leave that for you. So my last slide is that, you know, the take homes are to go to the patients, you know, tap into the existing communities. Don't necessarily try to start your own. You have to be inclusive and think actively about diversity before you launch. Work with patients before and during and after the study. You know, don't build something and then invite people in after the fact. And take feedback seriously and show it. And then be authentic. You know, try and engage with people within the community as often as you possibly can. This is our team led by Nick, and it has grown since this. And there are just countless people who have been part of this over the years, and I can't thank each one of them enough. And with that, I'll stop and I will take questions.
Well, thanks, Corrie, for what is always a truly inspirational and thought-provoking kind of approach to science and advocacy. We do have some questions. This is a question that I'm very happy to see. I was hoping that this might happen. The top ranked question right now is, how could computational biologists and bioinformaticians from this conference and community contribute?
I am so grateful to be presenting at this particular conference for that exact reason. We need people, we need computational biologists to help us across the spectrum of generating the data to making use of the data. So like I said, right now you can go, you know, the easiest way to take a, I think, let me tailor back. Let's start with where to begin. And so I think to begin, we would love for people to take a look at the data that's in the public domain and, you know, help us conceive of the best ways to use it. So we have data released now in metastatic breast cancer, angiosarcoma, and metastatic prostate cancer. You can find that data in cbioportal.org, or you can go to our master website at joincountmein.org and click on the individual projects. Each project will have a data tab, and those data tabs will both tell you all of the, like it hosts all of the methods for how we generated the data, as well as links to where that data is living. So step one is if you could go and take a look at what we have in the public domain and even just provide us feedback. Each project has its own info at mbcproject.org or info at ascproject.org. And all of us, our study staff, would love to get, you know, feedback from you as to just, what do you think? And where are the gaps? And what would be a good platform for us to host this data? We, like I alluded to, we want to be able to host it in research-specific portals. So are there other places we could house the data? And if we did, what are ways that would maximize the accessibility? So another way that I can envision is partnering up with people like at the postdoc level or in labs where there are no computational biologists, in order to help them understand what they can do with this data. And so really thinking about the utility of it, and then helping us think through how do we do outreach to people where we could match people with computational chops to labs and investigators who may be more clinical, so that those, the coupling of those can lead to a greater understanding of these diseases. So step one, take a look at what we have. Step two, contact us through our project websites and let us know what you think. I'd love to get like a working group together of computational biologists who are willing to like think about what we do with the data that's in the public domain. So thank you so much for asking that.
I'll just mention that the Waldron Lab has a particular project called CBioPortal Data that leverages the CBioPortal, the entire CBioPortal data set, and makes a bridge between that and Bioconductor. So getting started with this kind of thing with CBioPortal data is very straightforward. Clinical genomic studies at academic medical centers have a tendency to under-enroll minority populations among study subjects. Does the broad representation of hospitals and areas reached by CountMe and allow it to do better?
That is like, these are the top two questions I would hope would be asked of this. I really, whoever asked that question and whoever voted that up, I want to thank each and every one of you. This is something I'm passionately committed to, because we do skew young and we do skew white through our studies, despite the fact that we have reach into basically anybody who has access to the internet. I think in our project study staff thinks very much that we have a lot of work to do at the research side of this, building trust within communities that have been abused by the medical and research industries for hundreds of years. We have an entire team, sub-team within the CountMe and infrastructure that is focused on community outreach and engagement with like faith-based engagement with people in the community, to include barbershops and salons, engagement with young people at historically Black universities, HBSUs, in order to try to reach younger populations of people that may be willing to work with us in order to augment this message, and more so in order for us to gain trust within communities, so that not only our projects will improve in terms of representation and diversity, but other research initiatives can flourish as well. We're never going to get there if we don't really act together in a cohesive way to try and make this accessible to people who are in underrepresented communities. And not just these projects, but science and more generally, we absolutely have to do better. And if you have ideas beyond what I've said in this very short answer, again, please reach out to us, because we have an active subgroup within our projects that's their focus every day, is how do we reach out to underrepresented communities and how do we build trust. So I would love to hear your thoughts and ideas.
I think we have time for probably one more question. So I'm going to use moderator's discretion and jump down the list a little bit. Can you speak a little bit about how you navigate the balance between the need for patient privacy and the imperative to share data for research? And I might add to that how you incorporate patients' wishes in how you build things.
Absolutely. These are such excellent questions. So, it's step one, again, from our perspective, is talk to patients, talk to as many patients as you can. And then don't ever stop talking with patients. It's never done being built. You always have to re-engage, and you always have to re-engage new people. The whole ecosystem changes with time, and you have to make sure you're still doing right by people across the spectrum, from privacy concerns to maximizing their desire to share. So what we have to be conservative with respect to making sure we are protecting people's individual level data. It's just imperative to make sure that we protect as much as we can the ability to keep people anonymized. So we have a couple different tiers that where we can release the data. In the CBioPortal data sets, those are all somatic calls. So we're not releasing the raw underlying genomic data that can be used to fingerprint individuals. And we take away all of the HIPAA identifiers. And we take measures that would bin categories rather than specify, you know, a date of birth or even a year of birth. So instead of saying that this person was born in 1948 and developed metastatic cancer, in this year, we will start at the point when they were diagnosed with breast cancer and move everything relative to that. So you don't actually know when they were diagnosed, nor really, you know, how old they are now. And instead of saying that they're 48, they'll be in a bin from 45 to 50. And so with these different measures, aggregating the data, we strip the identifiers away and also try and, you know, bin categories to make it as anonymous as possible without taking away the ability to delve in and understand what's going on in a group that's 45 to 50 that may have, you know, it may have been 10 years since they were diagnosed. You just don't know when their birthday is, or, you know, other individual level information. And then when you get to the genomic data commons, which is, it has a DAC, it's more protected, that's where investigators can download the raw underlying genomics and concatenate that along with the other information we have in the public domain.
All right, well, it's been a pleasure. For those who are online, thanks so much for attending. Thank you, Corrie, for a fantastic talk. Remind your friends that this is recorded and it's worth a view. Corrie is an amazing person, has some amazing projects, and has really changed the way research is being done. So thank you so much. Take care, everybody.
Thank you. Thanks. Bye, everybody.